Hello everyone. Today I'm going to discuss about DNA replication. Since you all have revised our lesson on structure and functions of DNA and completed the recall activity, it will be easier for you to understand this lesson. So let's first see what is meant by DNA replication. It's the synthesis of new DNA molecules, which are called daughter DNA, from the information in parent DNA. Do you know why we should learn about DNA replication? One reason is that DNA replication is important for our growth and also for wound healing. If we consider about our growth, as you all know, we all started our life as a single cell called a zygote. But even a small child will have several billions of cells in its body. And if we compare the genetic makeup of her cells with the genetic makeup of the zygote from which she was developed, it will be identical. That's because DNA has replicated before each and every cell division and has transmitted it to the next generation of cells. Then if we consider about wound healing, once you pass out as dental surgeons, you all will create a lot of wounds during tooth extractions and while performing surgeries. You can do it without fear because you are 100% sure that the existing cells will replicate their DNA and transmit that information to the next generation of cells so that wound healing will take place effectively. Another reason for learning about DNA replication is that we can use DNA replication inhibitors to treat certain diseases. When DNA replication is inhibited, subsequent cell divisions and the growth of the organisms also can be inhibited. We can use DNA replication inhibitors as antibacterial drugs, antiviral drugs, and also as anti-cancer drugs. Now, let's look at another reason for learning about DNA replication. Do you know what's the most famous test that is performed on us today? That is PCR test for diagnosis of COVID-19 infections. And that is nothing but DNA replication outside the body. The term in vitro stands for outside the body. And these PCR tests can be used not only in diagnosis of diseases but also in vaccine production and many other techniques in recombinant DNA technology. So just like many other reactions taking place in our cells, DNA replication also is an enzyme catalyzed reaction. And the most important enzyme involved in DNA replication is DNA polymerase. There are different types of DNA polymerases in both prokaryotes and eukaryotes. In prokaryotes, the most important ones are DNA polymerase 1, 2 and 3. Out of the eukaryotic DNA polymerases, the most important ones are alpha, delta and epsilon. As the name implies, DNA polymerases should be capable of polymerizing deoxyribonucleotides into a polynucleotide chain. But if we take some deoxyribonucleotides and add the DNA polymerase enzyme along with the cofactor, that is magnesium required for this reaction, and provide the optimum temperature and the pH, still this enzyme will not be able to initiate DNA polymerization alone. If the DNA polymerases cannot function alone, what should they do? They should obviously get the support of a companion. And this companion is a DNA template to guide the synthesis of new DNA. Because DNA polymerases can polymerize deoxyribonucleotides only if their nitrogenous bases are complementary to the nitrogenous bases on a template. And as you all know, the DNA double helixes in our cells are very stable. They are stabilized by so many hydrogen bonds. But at the time of DNA replication, these hydrogen bonds will break down and the two polynucleotide chains in a DNA double helix will serve as templates for DNA polymerase. Unfortunately, it's very difficult to satisfy these DNA polymerases. They will not be happy with the template as a companion. They will ask for another companion, and it is a short segment of DNA or RNA which is called a primer. When the primer is there, DNA polymerases can start attaching deoxyribonucleotides to its 3' end. Well, now let's look at another interesting feature of DNA polymerases. That is, they have two catalytic sites with completely opposite functions. This site is responsible for making phosphodiester bonds and synthesize a new DNA molecule, and this site is responsible for breaking phosphodiester bonds and remove wrong nucleotides added to the new DNA molecule. This site is very important in maintaining a high degree of accuracy in newly synthesized DNA, because errors or mutations in DNA are detrimental to our life. Now, let me show you how the exonuclease activity of DNA polymerases help in maintaining the accuracy of DNA replication. As you can see here, this is a DNA polymerase enzyme currently engaged in DNA replication. So what is this molecule? It's a DNA template. And what is this molecule? It's a primer which is being currently elongated by the DNA polymerase enzyme. And this is the most recently added nucleotide to the growing polynucleotide chain. And at the moment, what is the location of it? Is it located within the polymerase site or is it located within the exonuclease site? It is located in the polymerase site of the DNA polymerase enzyme. Now what happened to the enzyme? It moved backwards. And where is the most recently added nucleotide now? Now it is located within the exonuclease site. What is the function of the exonuclease site? It is to check for the proper base pairing between the recently added nucleotide and the corresponding nucleotide on the template. Is it matching? Yes. Cytosine is complementary to guanine and we can see 3 hydrogen bonds there. So it is properly matched. Therefore, the enzyme will move forward and allow the next nucleotide to enter and form hydrogen bonds. Now what should happen? The enzyme should move backwards and allow the exonuclease site to check for the accuracy of the most recently added nucleotide. So is it accurate? No. Adenine is not complementary to cytosine. So what should happen now? The exonuclease site should use its exonuclease activity and hydrolyze the phosphodiester bonds and remove this mismatching nucleotide. Then what should happen? The enzyme will once again move forward and allow another nucleotide to enter into the polymerase site. So this is how the exonuclease activity of DNA polymerases help in maintaining the accuracy of DNA replication. Another important feature of DNA polymerases used in polymerase chain reactions is their thermostability. Within PCR machines, we have to use high temperatures as high as 95 degrees centigrades to break the hydrogen bonds and separate the DNA double helix, because they are the ones which will be used as templates for DNA replication. Within our cells, this is brought about by enzymes, but it is not feasible to use enzymes for in vitro DNA replication. Therefore, it's a must for us to use thermostable DNA polymerases in PCR. And generally, we use Taq polymerase isolated from a hot spring bacterium, Thermophilus aquaticus, for this purpose. In addition to DNA polymerases, many other factors are required for DNA replication within living cells, within our cells, within bacteria, within fungi, and some of these are enzymes like DNA helicase, DNA topoisomerases, primase, DNA ligases, apart from DNA polymerases. In addition, it needs several other factors like DNA template and deoxyribonucleotides, which we have discussed so far, and also single strand DNA binding proteins, ribonucleotides, and energy. So using all these factors, DNA will be replicated within living cells before every cell division during the synthetic phase of the cell cycle. Please use this link to watch a video on DNA replication in bacteria. You don't have to memorize the procedure of DNA replication from A to Z, but you will be able to understand how it actually takes place by looking at this video. And this knowledge will be important for you when you learn pharmacology and learn about how different pharmaceuticals are used to target DNA replication in bacteria, fungi, and also in our body. I'll stop for today and will come back to you with the second part of this lesson tomorrow. So goodbye for now and stay safe.